So I'm here from BeeBytes. We're a not-for-profit social enterprise based in Scotland, offering honeybee genetic testing services. And we're a combination of geneticists, statisticians, and beekeepers. So our aim is really to try and explain some of the complicated science and translate it into a way that's accessible and useful to beekeepers who are interested in these things on the ground, working with their own honeybees. So traditionally, we've had a lot of different analytical approaches to identifying the different subspecies of bee. The historical one that was always used was morphology or morphometry, which was looking at the physical characteristics of the bees, looking at wing shape, looking at wing veins. And over time, we've developed a wide range of DNA techniques, which John's obviously talked about. And that's really where the ability for us to really understand what's happening in populations has really taken off. There are lots of limitations to looking at physical attributes, but DNA gives us a much deeper understanding of what's going on. What has happened is that there's been a number of recent studies looking at population data, looking at the different levels of hybridization and the different subspecies in different parts of the world. And something we find is there's not actually been a lot of work in this area. Studies tend to be either very broad or they tend to be very looking at specific issues, because bee science is often very focused on a particular topic. But I'm going to hopefully address some of the most recent publications and identify areas where we do have some data. So one of the kind of earlier papers on this from 2018, which was done by my colleague Mark Barnett, along with Tim Reagan and David Ragg at Roslin Institute, was looking at the honeybee microbiome, which is really looking at the gut bacteria and the honeybee genetics to try and see if there were differences between these in different parts of the UK. And part of the results from that work was this chart showing a map of the UK and different samples. At the time, we talked about mitochondrial DNA. There were two separate populations of mitochondrial M lineage DNA identified, which is the gray and the black. So you can think of those as M lineage native bees compared to the yellow segments, which represent C lineage imported honeybees. And what we see from this data, although it was mostly focused in Scotland, is actually that there are areas where we have very high M lineage nuclear DNA, more so in the north and west of Scotland, more isolated areas where there's been less beekeeping, less imports going on. Whereas further south, we see more levels of introgression happening where there's obviously been more beekeeping, more imports. But interestingly, we start seeing here that virtually none of the samples, apart from there were two in Scotland, which are almost completely yellow. These were from commercial operations that were actively importing pure lineage bees. And on the west coast, we have Colonsay, which is a native honeybee conservation island, which is for M lineage. In almost every other case, we saw that there was hybridization, but there was genetics from both M and C lineage in different proportions. More recent study, which has been very useful to kind of bring us a bit more of an updated picture with a lot more samples. This is from Victoria Boswell, who's part of Mary's group, who was looking at, again, introgression in populations using whole genome sequencing. So this is looking at all the genes in honeybees rather than just looking at specific regions. And this really identified a similar pattern. So in this particular chart, the red is M lineage and the blue is C lineage. And again, we see a kind of similar picture here that, up in the north, we have, lots and lots of very pure samples here. Also, obviously, Ireland, which Grace will talk about later. But also down here in the southwest, we have some very pure M lineage. And then as we come further east and into the more central beekeeping areas, we start seeing more of the C lineage. But it gives us a similar kind of map of what's happening and is useful for us because it reflects in the same way similar results to what we saw in Mark's earlier work. What we've been now doing with BeeBytes is trying to look at ongoing data and keep updating things. So we're using a relatively simple tool for most of this analysis. It was developed by Alice Pinto, who will be talking this afternoon, or talking later on, which is looking at a small number of SNPs in the genome. So what that work basically did was to say, take samples of M lineage bees, that are known M lineage, take samples of C lineage bees, and look through them for a small number of markers, which are always set one way for M lineage and always set the other way for C lineage. And what we basically do is we analyze those small number of markers and we effectively add them up. And there's around about 100 markers. We basically say out of those 100 markers, if 70 look like M lineage and 30 look like C lineage, then we say that's a Q value of 0.7, so it's a 70% AMMB. It's a relatively simple test, and it's not going to tell you, if we sample a bee and say it's 95%, You can't immediately say, okay, everything about that bee is 95% AMM, but it's a strong indicator that that bee has low levels of hybridization. So this is a snapshot from a live map, which we're basically working on an interactive data portal. And this reflects, again, very similar data. We see very dark results. Obviously, we have Colonsay here, which is effectively pure, very dark black, but we also see very good results in Orkney, and then dark results in many areas, including quite heavily populated areas around the central belt. We then see other areas where there are samples which are obviously much paler. The scaling this is obviously difficult. I mean, even here, this sample here was 35% M lineage. So we haven't seen any really pure in the samples we've been testing, but then most people using our services wouldn't buy an Italian bee in and then immediately send us to us and say, is this a M lineage bee? So there's a bit of bias there. But we're seeing a similar pattern again, Scotland, and then again down here in the southwest, we're seeing darker, North Wales, we're seeing darker samples, central belt, central parts of the UK, we're seeing paler samples. Interestingly for us, we have this lovely section here, which is from the Devon project. This was a project organized through the B4 project. And with this, we looked, gone too far there. With the Devon project, we looked at 200 individual bees from a wide range of colonies across Devon. And this was to try and get a kind of county-wide snapshot. And what we found with this is when we did the analysis and we looked at the average level of M lineage was about 58%. So you might say, okay, well, that's 50-50, that's kind of hybridized, that's the end of things. But then what we did was we plotted out the admixture for each colony. So each column on this upper chart represents a colony. And what we found was really fascinating is that at one side, we had bees which are nearly 95% pure AMM. And at the other end of the scale, we have colonies which are only maybe 10% AMM, almost entirely pure imported C lineage. But there are colonies at kind of almost every ratio all the way along, which shows us that this is a really dynamic situation. You might think that, if you've had imports for, 150 years, 200 years, that it might have all kind of averaged out to a standard, but we're actually seeing huge differences in the population, and some bees are still very, very pure native genetics. Other bees are, have a lot of hybridization, and other bees are pure kind of imported bees. So for whatever reason, the populations aren't necessarily standardizing and leveling out. There's still a lot of movement going on in the genetics, which is quite interesting to see. So the Devon project was really useful for us because it gave us the snapshot, and we also obviously mapped it out looking at the actual geographical locations of these samples. And for example, we can see here we have a very dark spot. This was a colony which is one of these, up in the 90s here. And sort of almost like 5 miles down the road, we had one of the colonies here, which was one of the ones here. So we've got two beekeepers who are keeping bees maybe 5-10 miles apart. One of those populations has bees in it which are testing into the sort of 90% range AMM. The other population is testing down towards 10% AMM. So it's very indicative to us that these populations aren't kind of averaging out. There's interesting selection pressures, whether that's to do with how the bees are mating or environmental pressures are causing these populations to be kind of maintained. The other way, that way. So there are some limitations in the various methodologies that we're using here. The first one is that there are a lot of ways to look at this, a lot of ways to kind of crack the nut. And there are many different techniques, all of which have advantages and disadvantages. But the big issue is that they're often not comparable. So the SNP test that we're doing, we're looking at 100 SNPs and coming up with a very broad picture. Whereas if you're doing whole genome sequencing, you might be looking at hundreds of thousands of SNPs, and you get a much deeper picture. But there's costs there, and there's a different analysis you can do. So it can be quite hard to compare these different papers and say, okay, what are we actually seeing here? There's also the difference between different ways of sampling. You might sample single bees, or you might sample pools of bees. You might be looking at workers or drones. You might be analyzing queens directly. You might be combining samples from multiple colonies. So it's not always immediately comparable when you look at the data. But we're seeing general trends in terms of where we're seeing M lineage in the north and in the west. And that seems to hold up across all the methodologies, across all the different research approaches, which is a good sign. The second problem is granularity of the data. When you start looking into this, you get really excited because you find papers which are called things that, a European-wide study of native honeybees. You think fantastic. Then you drill down into the data and you look at it and they say, we sampled 500 colonies, and you think brilliant. And then you go, of which 8 were in the UK. And then you look at that and you say the UK average is 58%. But as we've just seen from Devon, that's not a way of really knowing what's happening in a particular country. And the aim of that study is to look at a European-wide picture. So it's very interesting research, but it doesn't really tell you what's happening in your region. And the sample size is really the problem that reflects things there. So the work that we've been doing with BeeBytes, the work we're doing in Devon, where we're really coming down to sort of postcode level, has been really insightful for us.
But what we can see is that the other story you hear a lot is, starting with Brother Adam saying, Isle of Wight disease, that, native bees were wiped out, they've all gone, we have to import because, there's no native genetics left. And yet when you look at the results that we're seeing here, pretty much every colony we test has some native genetics. We sampled Devon and more than 50% of the genetics in those bees was native. And in some cases, those bees are fairly pure near native bees. So the arguments about we need imports to increase diversity, we need imports because the local bee genetics are too weak or unsuitable in some way, are obviously not true, because on average, our bees are more native genetics than they are imported genetics, quite often very strongly so, despite how long we've been importing for and despite how much we're doing. And this really comes down to natural selection and what the pressures are on populations. We know that native genetics have been selected for local adaptations to do with the weather and the climate and the season and the forage. And those genes are just more naturally likely to survive because they're set up to survive. So no matter what we do with imports and things going on, we're always going to have populations that will maintain the genes that are important, and local genes are more likely to be important because if they weren't important, the bees in the UK would have lost them thousands of years ago because they weren't any use. So really, at this point, we're probably saying that we're not going to get a pure native bee population in the UK. We're not going to get rid of all the imported genes and never see it again. We've had hybridization. That's where we are now. But in terms of imports, what we need to do is potentially stop importing so that we kind of stop where we are, we deal with what we've got, and then we go forward from there. It's a slightly joking analogy that I've made before. You imagine if you're breeding dogs, for example, and you're breeding Alsatians. Alsatians were made out of other breeds of dogs hundreds of years ago, but now we have a definition of what an Alsatian is, and you're going forward with Alsatians every year. You don't every couple of years think, oh, I'll just put a Chihuahua in there or a Great Dane in there and just mix it up a bit and see what happens. But that's what we're doing with our population. The population is trying to improve naturally and trying to select itself in the best direction, and we just keep adding new genetics in to see what happens. And because they're open native, we have no control over it. So really at this point, there's lots of reasons to stop imports for pests and diseases and things, but we're working with a very mixed population that we could improve if we stopped kind of tinkering with it and just let it naturally go forward from where we are now. Final little thing, I'm running over very slightly, just to talk a little bit about feral colonies, because this is something that comes up quite a lot, and Andrew asked me to mention this. Something we discovered with feral colonies actually is that feral colonies are genetically extremely similar to local colonies. There's normally less than a 5% difference. So again, this mythical idea that somewhere out there are trees of bees that are completely unlike any of the imported bees that we're keeping at home isn't really true. Bees mate in the open, they all crossbreed. The bees that are living in trees are mating with the bees that are living in hives. The nest sites in trees are being repopulated with swarms from hives. And feral colonies are under a lot more pressure often because they have, they don't have beekeepers helping them necessarily. So if a colony is having starvation issues or having disease issues, there isn't a beekeeper to step in and prop them up. And because they reproduce through swarming, and the swarming is basically uncontrolled, they're not being split, those colonies are more likely to be undergoing kind of swarming, and swarming events are quite stressful for colonies. And so the survival rate when you swarm and try to set up a new nest are all lower. So that makes you think, well, feral colonies aren't any use and there's no point really having them. But actually, feral colonies have a lot of things going for them which are very important. They are being bombarded with all these selection pressures all the time by the environment with no help. So they're dealing with varroa, they're dealing with, trying to find nesting sites, trying to find forage, they're not being fed, they're not being treated, which means that every time a feral colony gets established, even if it's come from a swarm, it then suddenly gets a whole lot more pressure than it would normally get being kind of looked after by beekeepers. There's obviously pressures to do with finding nesting sites, and that's something we can look at working on. But the other advantage they have is significant drone production, that the comb in the colony is entirely at the whim of the bees, what they want to build. So feral colonies will always usually have more drone than in a managed colony because there's no one putting in worker comb and taking out and destroying the drone and saying, oh, I need to get rid of that because of varroa, or I don't want drone because it reduces my honey production. So having more drone means that if feral colonies are surviving these pressures, then the genes that are allowing them to survive are more likely to spread in the population because there are more drones coming out of those nests and transmitting. So while feral colonies right now are very similar to native populations, the more feral colonies we have and the more selection pressure, environmental pressures happen, the more likely that any evolution of resistance that we start seeing in populations may well come out of these, because as beekeepers, we take a lot of the pressure off our bees, which makes less incentive for them to have to evolve to survive these things. So that's a summary of really where we are just now, the UK beekeeping. I think we're about to have a break. Is that right, Andrew? No one else?